Hello, I am Inhyuk Song, a student at Busan National University. Thank you for giving me a great opportunity to present my talk. I'm going to talk about my research work for a deep learning approach for copy number variation detection using short-read sequencing data only. There are a lot of research papers that have been published about copy number variation associated with various diseases such as breast cancer, Alzheimer's disease, neurodevelopmental disorders, and esophageal cancer. This shows how significant copy number variation analyses are for human disease studies. What is copy number variation? Some genomic segments are deleted. Other segments are copied into a certain genomic position as illustrated in this figure. Copy numbers change significantly, especially in cancer cells, and copy number variation, abbreviated as CNV, is involved in the proliferation of cancer cells. CNV in cancer cells is specially called somatic copy number alternation, SCNA, which increases the expression of oncogenes related to proliferation or deletes tumor suppressor genes. According to a 2010 report in Nature, approximately 3,000 cancer samples were analyzed with 17% of cancer cell genomes amplified and 16% removed. Normal cell genomes are approximately 0.35% amplified and 0.1% deleted. How do we detect these copy number variations in the genome? One of most common approaches is based on read depth information. If we use read depth information from sequence alignment data, we can analyze the increase or decrease in sequencing depth compared to other genomic segments by comparing differences in depth of sequencing reads so that we can determine redundancies or defects of genomic segments. These redundancies and defects tell us copy number variations. As shown in the figure, we can see that much more reads are mapped in the area highlighted in red compared to its surrounding area. Usually, this copy number variation involves genomic segments ranging from several hundred base pairs to 5 mega base pairs. When we used short-read sequencing data only, it was quite limited to detect copy number variation. Since long-read sequencing technology emerged, we are able to determine such copy number variation a lot more accurately than before, as illustrated in papers published for recent years. Although long-read sequencing improved the detection of copy number variation, long-read sequencing itself costs a lot higher than short-read sequencing. In our study, we want to use copy number variation results performed by long-read sequencing as label data and to develop a deep learning model to determine CNVs using short-read sequencing data only. We obtained short-read sequencing data for NA1 to A78 from the Thousand Genomes project and used this as our input data. The gold standard CNV result for this NA1 to A78 genome sequence is available in DGV, database of genomic variants. We use this as our label data. This CNV label data result from long-read sequencing projects for NA12A78. NA12A78 is a human cell line commonly used for human genome variation studies. We mapped CNV label from DGV to sequence alignment data obtained by aligning the NA12A78 short-read sequencing data to the human reference. 20% of label data were used for training and the rest for validation. Detecting genome segments involved in CNVs is similar to outlier detection in one-dimensional signal data. In order to design a deep learning model, we started this project based on our previous work published for a peak detection model consisting of convolutional neural networks. This model called CNN peaks, and it mimics human visual inspection using convolutional neural networks. We used a reference sequence annotation binary vector and a chip sequence one-dimensional signal vector as input. We implemented a one-dimensional version of the GoogleNet architecture, which showed outstanding performance on ImageNet. We also applied advanced normalization techniques such as batch normalization to preserve the distribution of datasets as passing through multiple layers. This is performance validation for our chip-seq peak detection. In Figure A, for the GM12A78 cell line chip-seq data, while the sensitivity of CNN peaks was reduced by about 10% compared to other existing tools. However, our CNN peak significantly improved the specificity to almost 97%, while other methods were less than 76%. As a result, CNN peaks improved at least 8% over existing tools in terms of F1 scores. Unlike the GM12A78 cell line that is known to have a relatively normal karyotype, the genome of the K562 cell line has more complex karyotypes due to the frequent abnormal structural variations. For this reason, calling peaks in the K562 cell line data is more challenging than in the GM12A78 data. While the sensitivity of CNN peaks dropped it by almost 10% for the K562 chip-seq data, the specificity increased by 24% compared to the average of the other tools. This was quite successful to decrease false positive cause of chip-seq peaks. We modified the CNN peaks and designed our CNN architecture for CMB detection as illustrated in this figure. Here are some details of our new CNN model. First, we convert our raw data via data processing into input vectors for two CNN layers. These two CNN layers can reduce the number of trainable weights. Sequence alignment data in band format is transformed into a read depth profile through pre-processing as shown in the left figure, and the read depth profile vector is reduced via convolutional layers as illustrated in the right figure. This decreased the number of parameters that the model needs to learn through the pooling layer. Once the input vectors are processed via two CNN layers, they are trained through the residual unit, which is a core operation of our model. This residual unit can change the number of units into n, which is a hyperparameter. N units are stacked sequentially, the units as shown in the red square box. This residual network unit can improve sensitivity in a local area of the sequence. When a model is designed with deep layers, its optimization could be difficult. The residual network unit can resolve this issue via identity mapping. As shown in the left figure, downsampling with CNN can implement the identity mapping. This reduces the loss of information as data pass through layers. This residual networks has been validated in various applications as illustrated in the table. We included in the residual network unit in our CNN architecture. After all these processes finish, the cross-entropy loss of the predicted values versus the actual label data is calculated. Based on this cross-entropy loss, backpropagation updates the weights of this model as shown in the red square box of the figure. We compared our method with 10 existing applications for CNV detection. These 10 methods were selected based on its popularity in Zhang et al. 2019. This plot shows our preliminary results. We computed the number of bases that are labeled as CNVs. Comparing to actual bases labeled as CNVs, while NumPy predicts CNV bases 33 times more than actual CNV bases. Our model predicts 1.51 times more than the actual bases. We also measured precision, recall, and F1 scores based on the number bases involved in CNVs. As the performance of our model is shown in the rightmost side of bar plots, it shows high recall while relatively low precision. In terms of F1 scores, our model is the second best. In summary, we developed a convolutional neural network model for CNV detection using short-read sequencing data only. There is still some room to improve our CNN model. We plan to apply some techniques designed in the CNN-PIX pipeline for more precise CNV detection within a window. In addition, we are working on updating post-processing steps of our method to increase precision. We will also measure the performance of the CNV detection based on the number CNV events in addition to the number of bases involved in CNVs. Thank you for listening to my presentation.